I wanted to quickly ask if there is anybody who has not used the PDB in any capacity. And I think the quickest way to do that would be, do you want to raise your hands, or not used if you want to write on in the chat or something like that? Because I just want to see how much I should say in detail. Of course, I can't see the chat.
I can tell you, Shuchi.
Okay.
And so far, nobody's saying anything. So
Okay, so then
that is a very good sign.
yeah, so it seems like everybody has used the PDB in some form. But the point that we want to do in this, the main thing we want to do in this morning's, the first part of this morning's tutorials, is to enable you to find structures and understand what is contained in these structures. And so if you remember, yesterday we were talking about these articles that we are writing, they need to be substantiated with evidences from the literature, so papers that you will cite, but also evidences from the structures themselves to illustrate whatever claim you're making. And so in order to do that, you need to find structures in the PDB. And in the PDB, you probably already know that, if you search for hemoglobin, you'll get hundreds of structures. So you have to find that hemoglobin or those hemoglobin structures that help illustrate whatever point you're trying to make. So fetal hemoglobin bound to oxygen, carbon monoxide, whatever it is that you're trying to discuss. And so what I want to cover in the first part is sort of giving you a sense of how to navigate through the database. And then once you have identified the file or those files that you feel are relevant to your story, how to understand what is contained in those files and what in that structure will you focus on or illustrate or highlight or color in this and that color in order to tell your story. In the second part of the tutorial, we're actually going to look at a molecular visualization tool, MolStar in this case, and understand some conventions, which you may all be very familiar with, but just to make sure that everybody understands this, and because there were some students with different backgrounds. And then we will do a quick exercise in MolStar to get comfortable with just navigating through the structure to find the particular residues, chains, whatever it is that you're trying to illustrate. Okay, so the database is available with two entry portals. One is the rcsb.org portal, and the other is the PDB 101 rcsb.org portal. And the molecule of the month features are displayed on both pages. But what you get from the rcsb.org is the detailed listing of all the structures, whereas the PDB 101 gives you a more organized and annotated view of that same archive, but with descriptions and explanations that makes it less cumbersome and less overwhelming. So for this particular discussion, we are going to think about the rcsb.org portal. And so in that, I am going to, so this is what the rcsb.org page looks like. I'm actually surprised, David, that we are seeing the February molecule of the month.
I don't know what's up with that. I should go check.
Yeah, I was like, how did that happen? But go ahead. You were saying something, David?
Just everybody's getting a preview of next month.
Yeah, it seems like. So what you can do on this page is you can type your query in the top search box and begin your search. And this is where I wanted to point out that you can either search by typing in that text box a text. So that top search bar text is just a text search. Search is a text search, but then you can also search based on various other criteria and parameters. So you can have different kinds of attributes, like the details about the deposition, details about the chemical components, details about the protein, the nucleic acid, various other things, the experimental method, and so on. So you can search by attributes. You can search by sequence by providing one particular PDB ID and picking up the sequence from there, or you can just paste a sequence here. You can search by structure similarity and chemical. So there are various options possible. And that is what is sort of summarized in here, that you can do searches. And as an example, I just wanted to give you a quick sense of, so if you start writing hemoglobin, you will start getting these different options. So it's asking you, are you looking for uniprot molecule name hemoglobin? Are you looking for these keywords in the structure title, description, so on and so forth? And through this, you can narrow down your search result to something that is meaningful to whatever your question is, whatever your interest is. Okay. And you can also, once you have done that search, so let's say we do this search with hemoglobin, and notice that in hemoglobin, there is hemoglobin, but then hemoglobin A1 chain, alpha chain 1, alpha 1 chain. There are all these different possibilities. And if I click on one of these, it is going to only give me the uniprot molecule name with just that. So if, for example, if I just did hemoglobin, I get just 4 structures, and there is no human hemoglobin here. So this didn't really help me much. So let's try broadening the search. So in the human hemoglobin, if I tried, hemoglobin alpha, it's giving me this. So the point here is that you may have to play a little bit with these search options to find what you are really looking for. Okay, so let's say we go to this. In structure description, there is hemoglobin, and I click on that. Now notice there are 500 and something structures. There are 280 something Homo sapien or human hemoglobin. So let's click on that and look through this. So what I'm trying to do is trying to illustrate a scenario where you can get a large number of search results, and then by using various different parameters, you can refine that search result to get one or a few structures that are relevant to your particular story, to your particular question. So here, what you can do is once you see the search results, you can see that there are different ways you can refine it by the source organism, by what method was used for structure determination, whether it's a protein or protein nucleic acid structure, what resolution, when was this structure deposited or released, etc. And you can also do this refinement by creating different kinds of tabular reports or viewing the search result set in different ways. So immediately this stands out. This is something different. So you may want to go in and investigate if this is relevant to your query. All the other structures look similar. However, they may have different ligands bound, different pH, different conditions, so on and so forth. So having these different parameters to refine your result set gives you the ability to quickly narrow down your search result to find the few structures that you want to examine. This same refinement can also be done by opening this in the query. So you've done a query where your query has this phrase hemoglobin in this particular category. You can now add more parameters and conditions by clicking on this open in query builder and add conditions to get to your particular structure of interest. So what we wanted you to do as a sort of hands-on exercise is coming up shortly. But before we go there, I wanted to point out that in addition to querying, you also have the capability of browsing the archive, the entire archive by different annotations. For example, You can browse the archive by the source organism, by the enzyme classification, by whether or not it's a membrane protein, what CAT, SCOP, different criteria. So this ATC is different classes of drugs as classified by WHO. So these are different options where you can go through these trees, browsing trees, and identify that group of structures which have whatever criteria you are browsing by. And the exercise 1 is we wanted to find the earliest PDB structure of a human deoxyhemoglobin determined by X-ray with a resolution better than 2 angstroms. I'm going to pause for 2 minutes. And since all of you are familiar with the PDB, why don't you just open your browser and try it? I'm looking for a PDB ID. So is everybody able to log on to the PDB's website and do some quick browsing searching? Anybody not able to do so? And in the chat, I'm seeing Samuel says that you haven't played with the PDB before. So this may be a little new to you, but I hope it won't be too difficult. Sure. And so any questions? And maybe David, you can help me if there are questions coming up. Here there are questions. Oh, sorry. It is, yeah, www.rcsb.org. If you haven't figured that out. So did anybody find any PDB IDs that matches this criteria? Okay, Zach says 2HHB. Okay, so we are getting different answers. And this is something that I do want you to realize, that depending on how you implement your search criteria, the parameters that you define, you may come up with different structures. And sometimes even with the availability of these multiple structures, you're then faced with the choice of like, should you use this, should you use that? And then maybe we can pause for a quick moment and we can talk about how you make those decisions. So I'm going to go back to the website, and we had already done this hemoglobin search. So in this hemoglobin search, I've selected the human hemoglobins. That's what we wanted. We wanted a crystal structure, so I'm going to select that. And we wanted the structure to have better than 2. So I'm going to select these 2. And what else did we have in our question? Better than 2 angstroms, X-ray crystallography, deoxyhemoglobin. So we haven't identified that yet, but we've selected human. So let's see what we get in our. So after selecting all these boxes, if I click on this button, it has run that set. So now out of the 800 and something, we are with a smaller number, sorry, 200 and something, we're at 125. And what I can do is I wanted to find the oldest structure so I can sort it. And this is what I get. So someone had said 2-HHP. So notice 4-HHP, 3-HHP, and 2-HHP. All of these were they reference to the same article. They are released more or less at the same time. But if you look in their these structures, the R factors, the quality, they're slightly different. And so in this case, 4-HHP is what I had come up with, but 2-HHP is also acceptable. I want to pause a moment and ask Natalie and Helen if you had a reason why you chose 1A3N or 1HV4, or did you have any questions? Does anybody have any questions? You had deoxy in the search name. And so here, again, whether or not deoxy appears with a dash or with a space or, together with the word hemoglobin, that may also make a difference. And sometimes, the word deoxy may or may not be written. And yeah, okay.
Hey, Shuchi?
Yes.
This is Dave. When I was playing with this exercise, which is really super useful, it turns out it makes a difference, the order that you pick the refinements. And you can get in all kinds of unusual places depending on what order you do it.
Exactly. And so I think this is an opportune point to, time to talk about this point of how you do the search may impact on what search results you get. But then once you have these search results, you may come up with, like, let's say 20 structures. Of these 20 structures, when you're doing your molecule of the month illustration, you probably only need 1 or 2. You're comparing 2 structures. And so how do you get down that result set of 20 to 2? So you probably need to be guided by the story that you're trying to tell. So if you're looking for a particular mutation or a particular condition under which the molecule adopts a certain conformation, binds or does not bind to a ligand, or things like that. And so then the quality of the structure also is important. So when you are displaying your figure, you want to make sure that the molecule is as complete and as correct as possible. And if there is a particular region that you want to focus in and that region is not well resolved, you probably want to go and pick another structure, or make it clear in your article that this region was not resolved. David, do you want to say anything to that? How do you address these when you're selecting structures?
Yeah, just that when I'm looking for structures, I try to be very critical and try to do things 3 different ways and pick the answer that comes out the best, just because there are so many options for searching through the database, and so many criteria that you use to choose the best structure. So just be critical and try a bunch of things.
Okay, so with that, we are going to move forward. So I'm going to use the 4-HHP as the example and go ahead. And what I want to do is, so whether you choose 4-HHP or 2-HHP or any other structure, that's not what is important beyond this discussion. What I want you to be able to go ahead and do is, once you have chosen a structure or a few structures, how do you understand what is contained in that structure, and how will you then from that structure choose the appropriate chains or residues or ligands to illustrate in your article. So let's look at 4-HHP, and the next part of the slide was to sort of give you a sense of what that particular structure contains, and this is displayed in the structure summary page. And so let's look at the structure summary page. So here we are looking at the title, who solved the structure. This is one of the Perut structures. And we have a quick snapshot of what the molecule looks like, okay? Here we have a slider that gives you some sense of the quality of the structure. And in this slider, these bars are telling you about the quality of the structure. The bars, the more they are on the blue side, the better the quality. The more they are on the red side, the quality is not as good. But you have to remember that this structure is a really old structure. This is from the 1980s. And the standards that were used at that time for solving, so they didn't have all these fancy visualization software and computer analysis tools and so on and so forth at that time. And so I'm actually amazed that they got all these coordinates, organized to be able to understand what is going on in the structure to start with. And this has historical value. There are going to be issues and details. So there is a complete validation report that you can open and look at. But the takeaway point from this discussion here is that it may be valuable for you to look at the slider and understand something about the quality of the structures before you go ahead and choose that for your analysis or for your molecule of the month story. So here is a connection to the literature. So you can go to the PubMed and read the article, or here's the DOI, the digital object identifier for the publication describing the structure. In this section, you have information about the macromolecules. So notice that there are two different kinds of chains, the hemoglobin subunit alpha and subunit beta. And there are two copies of each of these different polymers. And so the four of these together, so the chains A, B, C, D, is what makes up your hemoglobin molecule. And so each of these types of proteins is called an entity. And the four of them coming together forms your assembly, the tetrameric assembly, that is going to function as hemoglobin. In addition to the macromolecules or the protein chains, there are small molecules. So in this case, of course, heme, because this is a hemoglobin structure. And there are phosphates. There may be ions, ligands, cofactors, ATP, other things that are present in the structure. In this case, there are only two different small molecules. And then there is more information about how the structure was solved and the details about the crystallography and the validation slider again. So this gives you a quick glimpse of what is contained in the PDB file. Now we can go ahead and, with this idea in our head, go forward and examine the 3D view, which we'll come to in a moment. But any questions so far about what is contained or what you can find on the structure summary page? No? Okay. So I'm going to go ahead and tell you a little bit about what actually the data file really looks like. So what you're seeing on the structure summary page is a description or a summary of the fact that this is what the overall shape of the molecule is. These are the different proteins and ligands, etc. But the actual file is, this is really what the PDB archives. The three-dimensional coordinates of every single atom that makes up that particular molecule, that particular structure. So here are your X, Y, and Z coordinates. In this case, we are looking at the valine, the N, C alpha, C. So every single atom in that residue has these coordinates. And that is really what the PDB is all about, the three-dimensional coordinates of experimentally determined structures. In addition, there are all these other metadata fields that are populated about who solved the structure, what this structure is all about, what ligands there are, all kinds of things. And this is really what the PDB file contains. Though more often than not, you probably will only interact with the PDB file in this format. And you may never ever open the PDB file. But the PDB file actually has also evolved in time. And so, well, I won't say evolved. This particular format is not the format that is currently promoted because there are many limitations in that. And so now we have transitioned to this PDBX or MMSIF format, which is more modern, more powerful. It is machine readable. And you need a little bit of practice to get oriented and figure out what this file is saying. But in detail, you can now get information about every single entity. So remember we looked at the hemoglobin alpha and beta chains, the heme, phosphate, waters, etc., the sequences of every single polymer chain, and all the ligands, all the coordinates of the polymer, the proteins, the ligands, waters, everything is included in explicit detail in this file. And I'm specifically pointing these things out because some of this terminology is what you will see when you look at the visualization tool, the Molstar visualization tool. You will see these terminologies over there. And that's why I'm making it a point to describe this here. Any questions? Everybody is good so far? Okay, so as a quick review, I wanted to go over this particular structure. So 2-HPS is the structure of the sickle cell hemoglobin. And so in the sickle cell hemoglobin, what happens is it's a hemoglobin structure. It's deoxyhemoglobin, but it is sickle cell, meaning it has that mutation. And so if you look at the deposited coordinates, the asymmetric unit, you actually see how that valine, which is mutated, sticks to the another hemoglobin molecule. And this is sort of the basis for formation of the fibers that then distort the red blood cells and so on. But without getting into that biology, what I wanted you to think about is that in this structure, in this entry, in this PDB entry, there are actually two hemoglobin molecules sort of stuck to each other, interacting with each other. And there are two assemblies. So each of these hemoglobin molecules is considered an assembly. Within each assembly, there are 4 chains of 2 types. So there are 2 entities and 2 copies of 2 entities. And then there are, of course, these ligands and waters and things that are. So in this case, there is only one small molecule ligand, the heme, and then there are waters. So this is what is summarized in this figure, where the entry is the whole thing. The biological assembly is each of these hemoglobin molecules. There are polymer entities that are these chains that are shown in orange and blue. Then there are non-polymer entities, the heme groups and the waters. And each of these entities, so the orange colored entities are the alpha chains and the gray ones are the beta chains. And then there is instance. So if you are looking at this particular chain of the alpha chain entity, you will call it the instance. So these are again terminologies that you may encounter when you're playing with Molstar, which is why I'm bringing them up. So is everybody okay with these words? Any confusion? I'm happy to review and explain.